Welcome. Today we are going to learn about the two hybrid assay or the two hybrid system. There are various methods to detect the protein-protein interaction. And from that, there is the bacterially expressed glutathione S transferase fused protein system. Then there is a fast display immunological studies and the two hybrid system or the two hybrid assay. These are the methods to detect the protein-protein interaction. Thus, the two hybrid assay or the two hybrid system, it is used for the detection of the protein-protein interaction. Now, by having these two hybrid assay, we can screen expression libraries for the novel interacting proteins. Expression libraries means what? The cDNA libraries. We can screen the cDNA libraries for the novel interacting proteins, or we may also study the protein-protein interaction of the known proteins. Like how the beta-galactosidase, the alpha peptide, it is interacting with the omega, and the functional beta-galactosidase, it is going to be formed. That it is called as the alpha complementation. This it is studied with the help of the two hybrid assay only. And the number of activators, the number of repressors, they are studied with the help of this two hybrid system. So here for having the two hybrid system or the two hybrid assay, there is a use of the activator. We know that the activator, it has the two domains. One, it is the DNA binding domain, and another one, it is the activating region. Okay, this it is the activating region, and this it is the DNA binding domain. DNA binding domain, it is interacting with the activating region. The complete activator, it is going to be formed, and then that activator, now it is able to interact with the DNA, and it is able to interact with the basal transcription apparatus for the expression of the gene. And these two domains, they are present on the two different proteins or the two different peptides, and that two different peptides, they are interacting to each other, and it is going to give you the complete activator. So for the two hybrid assay or the two hybrid system, there is a use of the DNA binding domain and activating region. Here for having the two hybrid system, the three constructs, they are made. Okay, on the plasmids, the three constructs, they are made. First construct, it is consisting of the enhancer, or as the host, it is the yeast. You may call it as an upstream activating sequence, and which is fused with the reporter gene. Okay, this it is the first hybrid construct, first plasmid consisting of the enhancer and the reporter gene. Here the reporter gene, it may be any one. The most frequently used, it is the LAGZ. Next one, it is hybrid construct, which is consisting of the activating domain of an activator, and here it is a prey. Prey means what? The protein which has to be analyzed. Okay, which has to be analyzed. The protein, the protein which is going to interact with the a particular protein. Okay, there like the one, it is the omega, and the second one, it is the alpha. Suppose you want to analyze the interaction of the alpha with the omega, then here the prey, it will be the alpha. That is, alpha, is it going to be interact with the omega or not? It has to be found out. So the alpha, it is going to be a prey, and to whom you will call as the bait. Bait, it will be the omega. So here the bait, it is the omega. That is the protein. Okay, the protein which is going to be a constant one. Okay, the protein which is allowed to interact with the number of proteins, that it is called as the bait. Like how the omega, it is going to be a bait, and that it is going to be fused with the DNA binding domain. Okay, so the third construct, it is a DNA binding domain gene. It is going to be fused with the protein which is called as the bait. Okay, okay, these are the three constructs which has to be made, and these are on the three different plasmids. Okay, now here the hybrid proteins, they are going to be made from the two hybrid construct. Okay, first one, it is a protein 1 fused with the activating region, and a protein 2 which is fused with the DNA binding domain. If the protein 1 and a protein 2, they are interacting to each other. Okay, if protein 1 it is interacting with the protein 2, like how the alpha peptide it is interacting with the omega. Okay, at that time there will be the complete activator, it is going to be there. How? Here it is the DNA binding domain. Okay, DNA binding domain, it is fused with the protein 2, which is a bait. Okay, here it is a protein 1, which is a prey, and here it is a activating region. Okay, means what? Anyhow, the DNA binding domain, it is interacting with the activating domain. Okay, activating region and the DNA binding domain, they are connected to each other via the proteins, which are the bait and which is the prey. Okay, and when this activating activity of the activator, it is going to be completed when the protein 1 it is interacting with the protein 2. And if it is not interacting, okay, if it is not interacting, then this activator it is not going to be completed. Now, how the experiment it is going to be done? Okay, now here the case 1. Okay, in the case 1, the here this one, okay, this one it is the hybrid construct which is consisting of the reporter gene and the enhancer. And in that, okay, in the case 1, we are also adding the construct which is consisting of whom? Which is consisting of the hybrid construct with the DNA binding domain and the bait. Okay, this DNA binding domain it is going to interact with the enhancer. Okay, DNA binding domain it is going to interact with the enhancer, but as there is a no activating region, okay, as there is a no activating region, the activating region it cannot interact with whom? It cannot interact with this transcription factor. Okay, so here this interaction it is going to be missed. Okay, the activating region interacting with the basal transcription apparatus, okay, and allowing the RNA polymerases to get loaded and allowing the transcription. This activity it is going to be missed when there is a no activating region. That is only the DNA binding domain, it is not sufficient for the activation of the gene. Okay, so in the case 1 you will find that there is a no expression. In the case 2, okay, the hybrid construct with the reporter gene and the enhancer it is there, and the second hybrid construct it is added, and it is of whom? The activating domain and the prey. As the yeast host, it is not consisting of the DNA binding domain. Okay, this it cannot interact with the basal transcription apparatus, although it is going to interact with the basal transcription apparatus. Okay, or it is interacting with the transcription apparatus, it is not able to load the RNA polymerases over the DNA, as the DNA binding domain it is missing. Okay, DNA binding domain, what is its function? Its function it is to recognize a specific gene, okay, and bind at that particular specific gene. But here in the case 2, what is missing? The DNA binding domain it is missing, so there will be what? There will be no expression. Okay, there is no expression. In the third case, okay, in the third case here there is the plasmid construct. All the 3 plasmid constructs they are present into the yeast host. That is a hybrid construct with the reporter gene and the enhancer hybrid construct with whom? Hybrid construct with the DNA binding domain with the bait, the hybrid construct with the activating region with the prey. And here if the protein 1 it is interacting with the protein 2, that is the prey it is interacting with the bait, then what will happen? What will happen? There will be the expression. And if you are, okay, if you are getting the expression of the reporter gene, if you are getting the expression of the reporter gene, then what you can say? That the bait and the prey, they are interacting to each other. That is, there is a presence of the protein-protein interaction between the protein 1 and the protein 2. And instead of this expression, if you got no expression, okay, you find out that the reporter gene it is not expressing, At that time, it is concluded that, okay, at that time, it is going to be concluded that there is no interaction between the prey and the bait. Okay, the prey, it is not able to interact with the bait when there is no expression. But when there is expression, there is protein-protein interaction. I think you have cleared the idea about the two hybrid assay. Okay. Why it is called as a two hybrid? Because the two hybrids, they are used here. Which are the two hybrids? Okay. These are the hybrid construct one, which is consisting of the DNA binding domain with the bait, and the hybrid construct activating domain with the prey. Okay. So it is called as a two hybrid assay. Okay. Two hybrids, they are used. Okay. So it is two hybrid assay. Okay, so if you like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. Thank you.